and I wanted you guys to be exposed to this first. I've changed the order of the lectures. There's several different pathways. This is a block outline for the entire block. We're going to talk about the mevalonate pathway, that's cholesterol synthesis. This is one of the other lipid pathways that was mentioned in the last block. And then we're going to talk about steroidogenesis. We know steroid hormones are very important. We're going to talk about how, where they're synthesized, and if there's any deficiencies and enzymes involved in that pathway, what can occur. We talk about catecholamine synthesis. Catecholamines are our neurotransmitters that are involved in our fight or flight response. And then heme synthesis and degradation, and then the blood clotting cascade. All right. So, starting with cholesterol synthesis. Oh, one thing I forgot to mention. So at the bottom, make sure you guys know all of the full names of the molecules. So there was some questions about abbreviations and knowing the full names during the exam. So make sure you know the full names because the abbreviations may not be present. You guys did great. You guys did awesome. So you knew it. You didn't think you did. Some people are like, no, I didn't, but you did. So for cholesterol synthesis, we start with acetyl-CoA. You know that famous acetyl-CoA. As I mentioned in the last block, all the lipid pathways use or produce acetyl-CoA. So acetyl-CoA plus acetyl-CoA come together to form hydroxymethyl-glural-CoA, and that is catalyzed by HMG-CoA synthase. HMG-CoA is then converted to mevalonate by HMG-CoA reductase. This is the rate-limiting step of this reaction, and statins target this enzyme. Statins work by competitively inhibiting HMG-CoA reductase. Now, do you guys remember from block 1? If you don't, it's okay, but what it means when something is competitively inhibiting an enzyme? What are some characteristics, in this case, of the drug? Yes? Correct. So your fellow scholar said it binds to the active site. So it binds to the same site as HMG-CoA reductase. What else? Say again? Vmax, wow. Yes, yeah. Excellent. That just gave me chills. Awesome. Listen, don't get me to pass out. Listen, I wasn't expecting that response. What else? One thing I'm looking for. Everything has been right.
Allosteric.
That just woke me up.
Allosteric.
Allosteric.
Where it doesn't change. It doesn't change the substrate.
No, the substrate does not change. Yep, the substrate does not change during this reaction. I'm looking for a particular thing. How can I phrase it? So this drug, what does it look like? It looks like the substrate. So, y'all said a whole bunch of stuff that I wasn't expecting. Y'all just really moved me. That's awesome. But it looks like the substrate. That's what makes it a competitive inhibitor of this pathway. So what statin drugs do, they bind to the HMG-CoA reductase and they compete with HMG-CoA, and so therefore they block the rest of this pathway, so mevalonate won't be produced, and of course you won't get cholesterol. Okay, does that make sense? Good job for now. And here is the key steps, what I just went over. Some other things that I didn't mention. This process, this pathway occurs in the cytosol, liver cells. It uses NADPH, and this HMG-CoA reductase enzyme, it is in the cytosol, but it's anchored to the smooth endoplasmic reticulum, but the active component is found in the cytosol. this enzyme HMG-CoA reductase must be dephosphorylated in order to be active. And some molecules that regulate its dephosphorylation is insulin and thyroxine, and then glucagon, epinephrine, and increased levels of cholesterol inhibit the dephosphorylation. Any questions so far? And so this is the hormone control or hormone regulation of cholesterol synthesis. So here is the HMG-CoA reductase. In its inactive state, you see it's phosphorylated. So what happens is if insulin or T4 is present or increased, that's going to cause the dephosphorylation of HMG-CoA reductase, which will cause the production of cholesterol shown here. Glucagon, epinephrine, and high levels of cholesterol, in addition to AMP, will reduce the dephosphorylations, or will cause HMG-CoA to be phosphorylated, rendering it inactive. Okay, so we've already talked about several regulators of HMG-CoA reductase. Another important regulator is this right here, sterol regulatory element binding protein 2. What this does, this is a transcription factor. When cholesterol levels are low, this will cause this element to be cleaved from the Golgi apparatus. It will travel to the nucleus, causing transcription increase and translation of more HMG-CoA reductase. Conversely, if cholesterol is high, it's going to inhibit the cleavage of sterol regulatory element binding protein from the Golgi, so it's going to be inactive. Does that make sense? Yeah, sorry. So, sterol regulatory element binding protein, it's found attached to the Golgi apparatus here. When cholesterol levels are low, that's going to stimulate this element's cleavage from the Golgi apparatus. This transcription factor will then travel into the nucleus and basically cause the increase of messenger RNA that codes for HMG-CoA reductase. All right?
So activation of the mevalonate pathway starts on kind of the right-hand side of the diagram then?
This is the mevalonate pathway here, and this is the HMG-CoA reductase enzyme. So this is just the HMG-CoA reductase enzyme. So it's phosphorylated, it's not active. When it gets dephosphorylated, it becomes active. So this element regulates the amount, the sterol regulatory element binding protein also helps regulate the amount of HMG-CoA reductase that's produced. Does that make sense? Any questions about this? So what are the regulators of HMG-CoA reductase? First one, cholesterol. Yep, cholesterol. What else? Epinephrine. Yep. So what else you guys said on this side? Glucagon. Yep. Insulin. Yep. AMP. Yep. What else? Say again. Thyroxine. Yep. And SREBP2. Questions? So we've already talked about statins and what they do. They are structurally similar to HMG-CoA, hydroxymethyl-CoA, and they're reversible. If a drug, the name will have statin in it, so you can see some of the examples shown here. These drugs are metabolized by cytochrome P453A4 and 2C9, depending on the statins. You don't need to know which ones metabolize which, just know that these drugs are metabolized by these two enzymes. And of course, they block mevalonate from being formed, and they decrease plasma cholesterol. So this is used for people with cardiovascular issues and hypercholesterolemia. Now their side effects will be muscle pain, weakness, and in severe cases, muscle breakdown. This muscle pain and fatigue seen is due to cholesterol is also responsible for making CoQ. You guys remember what CoQ is? Electron transport chain. Right. So if since it's involved in making CoQ, if there's decreased CoQ, then that means we're going to have less energy, right? That makes sense. So that's why you see the less energy, less ATP. So that's why you see the muscle weakness in this condition. And then in severe cases, there's going to be muscle breakdown because the cholesterol is also involved in membrane integrity. So that muscle breakdown will cause proteins to be leached from the cell. Myoglobin will be found in the urine, and that is rhabdomyolysis. Questions?
So what did you say those?
Just to be clear,
Go ahead.
side effects are from the statins.
Yes, sorry, the side effects are from excess statins. Good question. Yes. He didn't see your hand.
Yeah, I'll let it slide. So the CYP3A4 and the other one, which ones are those inhibiting? Are they inhibiting the ones above it, or like what were those doing? That made it important.
These drugs are broken down by these enzymes.
Okay.
You said what was responsible for creating CoQ?
Cholesterol. Cholesterol is involved in that. So you have decreased cholesterol, which you will have with statin drugs. And if you have too little cholesterol, then you might see muscle weakness. And that's because one of the reasons is because CoQ is decreased, and so not able to produce the amount of energy you need. Yes.
So the CYP enzymes, they actually metabolize the statins.
Say again.
The CYP enzymes actually metabolize the statins.
Yes, they break down the statins.
Is that those largely in the liver, or is that?
Yes. Yep. Any other questions? All right. So when you guys are treating, oh, sorry.
Never mind, it's right there. Go to the next slide.
Oh, okay. Oops.
Yeah, should wait like 10 seconds.
No, it's okay. So when you guys are getting your patient histories and the event you might have to put them on statins, you also need to ask, in addition to what drugs are you taking, you might need to ask what they're consuming as food sources or supplementation, because there are a number of food and supplements that affect 3A4 levels, and also, for example, red rice yeast, which is shown in that picture above, that is structurally similar to a statin. So what that's going to do is that if you give a patient statin and they're eating or taking the red rice yeast, it's gonna stimulate them having more statins. So that's gonna be more of a negative side effects with muscle weakness and muscle breakdown. So same thing for grapefruit juice. So grapefruit juice and red rice yeast are having the same effect. But what grapefruit juice does, it decreases cytochrome 3A4 enzyme. So because grapefruit juice is lowering that enzyme, you're going to have more statins in the blood, and it's not going to be able to be metabolized because the enzyme levels are low. So more statins in the blood, more potential side effects. Does that make sense? Now, St. John's wort, which is, yes.
So theoretically, could you prescribe a lower dose if you were to say, take the same combination with substrate?
That's a good question. I'm not sure because I don't know what test you would need to do. Maybe you might want to start with that, but you don't know how much your enzyme levels are being affected. So that might be a consideration depending on the severity of their conditions and if you don't have any other ways to treat them. So yes. St. John's wort is used to help with mood, increase mood. But St. John's wort does the opposite. It increases 3A4. So what's going to happen if you are taking that, if your patients are taking that and you prescribe them statins, they're going to have less statins in the body because there's more 3A4. It's breaking down the statins faster, so the statins are not able to produce the intended therapeutic effect. So that your patients are still going to have a cardiovascular risk. So those are important considerations when prescribing statins to your patients. Questions? All right, so humans, we don't have the metabolic pathways to break down cholesterol, so what we do is we excrete a little bit of it unchanged in our feces, and the rest we convert to other molecules that our body uses. And one of those that are very important are bile salts and bile acids. So we know bile salts help with absorption and metabolism. Bile acids help with absorption and metabolism of fats. So bile acids are produced, when they're produced by cholesterol, you see that cholesterol 4-ring structure here. What happens is when they become bile salts, hydroxyl groups are added at certain points on that 4-ring structure, and in the B-ring structure, that double bond is reduced. So there are two main bile acids that the body produces, and that is cholic acid here and chenodeoxycholic acid here. So cholic acid has 3 hydroxyl groups, it's a triol. Chenodeoxycholic acid has 2, it's a diol. So that is a bile salt. Now, bile salt is only this portion. Now, when you conjugate, a bile acid is only this portion, circled here. When you conjugate a bile acid with taurine or glycine, taurine is an amino acid derivative that is related to, or that comes from cysteine metabolism. When you conjugate them, you're making these bile acids more water soluble and they become bile salts. So before they're conjugated, bile acids, when you add glycine or taurine, this whole structure, this entire structure becomes a bile salt. Any questions? Here's just the key steps in the pathway of bile acid synthesis, and also bile salt synthesis. Remember, when a bile acid is conjugated, that produces a bile salt. All right. And again, the pathway for bile acid and bile salt synthesis. So in addition to making bile acid and bile salts, cholesterol can also be used to synthesize steroid hormones. And so we're going to talk about 3 different types: the mineralocorticoids, which is aldosterone, glucocorticoids, cortisol, and the sex hormones, androgens and estrogens. And so the synthesis of these steroids occur in different areas of the adrenal gland, particularly the adrenal cortex, shown here. There's 3 areas. In the zona reticularis, which is the innermost area next to the adrenal medulla. So in the zona reticularis, that's where our sex hormones are produced. In the zona fasciculata here, that's where cortisol is produced. And in the zona glomerulosa, that's where our mineralocorticoids or aldosterone is produced. So, there's a lot of stuff on this slide, but we'll walk through it and I'll highlight the key steps. Don't be scared. Don't be scared. So we know cholesterol is the precursor to all steroid hormones. It's going to be the starting point. So if we were to synthesize aldosterone, which occurs in the glomerulosa, what happens is P450 side chain cleavage enzyme will convert cholesterol to pregnenolone. Pregnenolone will be converted to progesterone. Then 21 alpha hydroxylase or 21 hydroxylase, same thing, will convert that intermediate to deoxycorticosterol. 11 beta hydroxylase will convert, deoxy is also called DOC. DOC will be converted to corticosterol by 11 beta hydroxylase, and then some other enzymes will convert corticosterol to finally aldosterone. Okay? If we want cortisol, what's going to happen is that pregnenolone, yes.
Sorry, sir. So all these reactions not reversible? Because the arrow is just pointing one way.
That's a good question. I'm not sure if they're reversible. I don't know if they're reversible or not. That's a good question. Any other questions? Yes. No. That's a good question. Not one that you guys need to know. Important. So if we are looking to synthesize cortisol in the zona fasciculata, pregnenolone or progesterone will be converted to intermediates 17-hydroxypregnenolone or 17-hydroxyprogesterone by 17-alpha hydroxylase. Okay? And then those intermediates will be converted to deoxycortisol by 21-alpha hydroxylase, and then finally cortisol by 11 beta hydroxylase. Any questions? Yes.
What's the 450 star protein?
That's the side chain cleavage enzyme. Yeah. So it's the enzyme that converts cholesterol to pregnenolone. Yes.
You didn't talk about what's made in the zona reticularis, and I was also wondering the significance of DHEA being highlighted up there.
Yeah, I'm getting to that. We're going step by step with aldosterone, cortisol, and then finally we are here to the zona reticularis. So zona reticularis, that's where our sex hormones are made. So what happens is that those intermediates formed in the zona fasciculata, the pregnenolone, progesterone, will be converted to androstenedione or dehydroepiandrosterone by 1720 ligase. So this enzyme gets you from the cortisol production to sex hormone production. Okay, so then these intermediates will produce testosterone or through aromatase enzyme produce estrogen hormones. So what's significant about aromatase? Aromatase converts androgens to estrogens. This enzyme is very important because what they found by doing research is that there are what's called estrogen positive receptors in breast cancer and postmenopausal women. And so what happens is if they develop drugs to block aromatase, so these aromatase inhibitors will block the formation of estrogen, So that will decrease or even stop the cancers from growing in estrogen positive receptors. Because what's happening is that excess estrogen is binding to the receptors, causing cell proliferation of cancerous cells. So we block that through aromatase inhibitors, which have the suffix ozol, anastrozole, and letrozole. So those are used to treat estrogen receptor positive breast cancers in postmenopausal women. And also in men, it's used to decrease gynecomastia. Now, there are times when we have excess cortisol. You guys probably may be experiencing that because of the stress you're under. Now, when you get old like me, it's going to make you gain weight. It's going to make you be irritable. So when I was in my postdoc, I was like, I gained a lot of weight. I was having a lot of health issues. And so my doctor recommended I take DHEA, which is this controversial, but it's thought to help with mood. And so what it does is that, like the theory is you have excess cortisol. Cortisol is responsible for glucose metabolism. So it's gonna make you take glucose in your cell and it's gonna cause gluconeogenesis to occur. So that cortisol is associated with weight gain and stress levels because of release of neurotransmitters. So what the doctor recommended is that I might consider taking DHEA because that's gonna basically increase more testosterone production for me, and then it's going to stop or counterbalance the effects of that increased cortisol. So that's what it's used for, and they call the condition adrenal fatigue, where they feel like you have too much cortisol. Have you guys heard of adrenal fatigue? No. Yeah, this some of you have. Yeah, I don't know. Like I said, it's controversial. Some providers, some don't believe that it's a real thing, but DHEA has been used to help treat it, and it's shown some anecdotal benefit for people that have used it. So, any questions about this pathway? Yes. Which one? Yeah, it's 21. And it's rated 21 hydroxylase and 21 alpha hydroxylase. So just in case you saw it two different ways, it's the same thing. Yeah. So thank you. That was a great segue question. So 21 alpha hydroxylase, you see they're shown in both the glomerulosa and fasciculata, as is 11 beta hydroxylase. So these two enzymes are very important. In addition to 17 alpha hydroxylase, you've seen it shown, it's also shown in two places. So, if these enzymes are deficient, one or more of them, you have a condition, what is called congenital adrenal hyperplasia. And so this is just the key steps to that pathway, highlighting the most important enzymes that you guys need to know. So, congenital adrenal hyperplasia is a genetic condition where one of these enzymes are deficient. So, these deficiencies are going to affect how the hormones that are made in the adrenal gland are produced. So, and we're going to walk through this table with the pathway. Now, so let's take 21 alpha hydroxylase. So I've blocked off the enzymes. 21 alpha hydroxylase is here, 11 beta hydroxylase is here, 17 alpha hydroxylase is here. Okay, you guys see that? Makes sense? So if we are deficient in 21 alpha hydroxylase right here, that means we're going to have a reduction in aldosterone, right? That makes sense? Right? Good, because it's stopping right here so you can't produce aldosterone. It's also here, right? So you're gonna have a reduction in cortisol as well, right? That makes sense? Yeah. So, since it's shunting the pathway this direction, you're gonna have an increase in sex hormones. That makes sense? Okay. Now for 17 alpha hydroxylase, that's here. So for 17 alpha hydroxylase, when it's deficient, you're gonna have an increase in aldosterone or mineralocorticoids, right? That makes sense because it's shunting the pathway this direction, right? For cortisol, cortisol levels are going to be decreased because the pathway is going in this direction instead of this direction. And then, of course, also sex hormones will be decreased. Is that clear? Does that make sense? So for 11 beta hydroxylase deficiency, we're going to start with the easy ones first. So here's 11 beta hydroxylase right here and here. It's blocking this. So we're going to have decreased cortisol levels, right? Then we're going to have an increase in sex hormones because it's pushing the pathway this direction, right? But we see an increase in aldosterone. Does that make sense? No, it doesn't. Right. It does not make sense. So the reason why this is happening is because of the pituitary gland. And so the pituitary gland senses a decrease in cortisol. And what's happening is it's going to cause the release of adrenocorticotropic hormone, ACTH. That is going to stimulate this entire pathway with the goal of producing more cortisol. But what happens is that we know we're not going to get cortisol because this enzyme is deficient. So we're going to get a bottleneck of intermediates, namely DOC, deoxycorticosterol. This intermediate has strong mineralocorticoid activity. So basically, this intermediate is going to be producing excess, and it's going to be doing the same thing that aldosterone is doing. So it's going to simulate or mimic the effects of aldosterone, producing an increase in mineralocorticoid activity. Does that make sense? A little bit? Yes.
I have two questions. So, with the 21 alpha hydroxylase, deficiency, for example.
Can you speak up? I can't hear you.
Yep, my apologies, sir. So, with 21 alpha hydroxylase, we're getting a decrease in mineralocorticoids and glucocorticoids, but an increase in sex hormones basically because all the substrate is being redirected.
Yeah,
Is that
it's going to the right. It's going to the it's going to the right. Yep.
So then my second is with the 11 beta hydroxylase deficiency. The doc is basically it's not a true one-for-one standard for aldosterone, but it's close enough that you're getting that.
Yes, that's a good way. Yes, that's a good way to put it. It's not necessarily it's not necessarily as strong as aldosterone, but it is strong, and because there's so much of it, it's producing the effects of more mineralocorticoids. Does that make sense? Yes.
Would that cause like hypernatremia?
Yes. Yep. Good question. Absolutely. Any other questions about this pathway? So what enzymes are involved in congenital adrenal hyperplasia? 17 alpha hydroxylase. Yep. Yep. Thank you. Any questions? Yes.
So I noticed that we bringing up the ones that duplicate themselves, but we haven't talked about 1720 lyase. And that one duplicates itself as well.
We talked about it in the sense that it gets us to the zona reticularis to produce sex hormones.
Right.
But because I don't mention it further, don't worry about it.
Oh, thank you. Thank you, sir.
Great question. Any other questions? So there's a lot of enzymes I didn't really talk about, right? So don't worry about those. Right? Make sense? So we're still in the adrenal gland, but now we move to the adrenal medulla, and this is where catecholamines are synthesized. We know catecholamines are hormones that are involved in our fight-flight response. So we start with the consumption of phenylalanine, which is what type of amino acid? Essential, yes. So we have to consume it in our diet. And it's converted to tyrosine by phenylalanine hydroxylase. That tyrosine can, through another pathway, be used to produce thyroxine or T4, or tyrosine can be converted to dihydroxyphenylalanine or DOPA by tyrosine hydroxylase, which is the rate-limiting step of catecholamine synthesis. In melanocytes, this DOPA, in the presence of tyrosinase, will make melanin, or dopamine, or DOPA, with dopamine decarboxylase can produce dopamine, which will then go on to produce norepinephrine, and then epinephrine. Epinephrine synthesis uses SAM, acetyldenazomethionine. We're going to talk about that. Acetyldenazomethionine is a major methyl group donor for the body, and we'll talk about that later on in this lecture. Also, this tyrosine can go on to produce homogentisic acid, which, in the presence of homogentisic oxidase here, will be converted to a number of intermediates that become fumarate. Where do we see fumarate?
Sure.
TCA cycle and urea cycle. So fumarate was an intermediate of the TCA cycle, and it was a byproduct of the urea cycle, right? Okay. And here are the major steps in that pathway that we just discussed. Now, when we have deficiencies in catecholamine enzymes, of course, they're going to cause disorders. We've talked about this and all of this. And of course, like I told you, they love phenylketonuria. So, phenylketonuria we know is a deficient phenylalanine hydroxylase enzyme. So, what does this deficiency cause? Somebody say it. I can't hear one person. Ammonia or close. You guys are in the urea cycle. It's a toxic. If you're right in that, it's toxic.
Acetyl-CoA. Acetyl-CoA.
Phenoacetate, phenolactate. It's still doing the same thing to the brain. It's toxic to the brain. But that's what happens. And you guys remember what type of enzyme does that?
Oh, transaminase.
Transaminase, yes. Y'all all remember that? Yeah, thank you. I don't expect you guys to remember that, but it's great that you know that if you do. So we talked about phenylketonuria. We're going to touch on it a little bit more. But when there's a deficiency in tyrosinase, there's albinism. We talked about albinism in the first block, some hallmark symptoms of that. And then when there's a deficient homogentisic oxidase, alkaptonuria, will result. We're going to talk about that. Now, dopamine decarboxylase, we're not talking about it here in the context of deficiency, but it is the target of a drug, carbidopa, which is used to treat Parkinson's disease. So what happens is, typically carbidopa and levodopa are given together. Carbidopa will block the dopa decarboxylase peripherally, and that allowing the levodopa to go to cross the blood-brain barrier without being broken down, and so that levodopa will be converted to dopamine, and then it will help alleviate some of the effects of Parkinson's disease, like the slow movements and the tremors and shakes. All right. So phenylketonuria, we know what that is. It's phenylketones build up. They are musty smelling, and they cause mental disabilities. So of course you guys know how to treat that. We talked about it like 3 or 4 times already. In terms of albinism, we talked about this. We know that they're basically melanin is not being produced, so that's going to cause your patients to have light skin, light eyes. Also, melanin is involved in optic nerve development, so they're going to be, your patients will be sensitive to light. So there's no real known treatment, but you want to just make sure that your patients have complete protection of their skin and their eyes. Right? Questions about albinism? Alkaptonuria is a new one. So what happens in alkaptonuria, there is a buildup of homogentisic acid because the enzyme homogentisic oxidase is deficient. That acid is going to cause 2 major symptoms. I have 3 in your slides. My apologies, I just caught that error. But it's going to be increased homogentisic acid in the urine, which will turn dark when exposed to air and light, as shown here. Also, this homogentisic acid, or HGA, it will deposit in connective tissue, causing ochronosis, as shown here. And so this condition is not really life-threatening or like debilitating in the sense of like some of the other conditions like phenylketonuria, but this ochronosis will cause significant arthritis that will develop when a person reaches middle age. And so that could be very severe for that patient, but otherwise the patient functions healthily and doesn't have really any major issues. And you, of course, would probably want to try to restrict their diet or limit their diet with amino acids that feed into this pathway tirelessly. Any questions about alkaptonuria? All right, you guys can take a 10-minute break.
Question for you, sir.
That doesn't mean. Yeah, but you never know if the slides are like how much you can slides each.
Yeah.
Oh.
Do my best. Turn into a 50. You good over there?
So can we get some of that, what is it called here? I need, is it prescription? DHEA. You know what? I can feel myself just like.
Oh, I never. I feel like you know.
Disgusting. My parents saw it, they were like, what? How is it that bad? It didn't help.
I know.
Oh yeah.
He said bile acid becomes bile salt when conjugated with glycine. So I said, this is glycine, so why is this still bile acid? He said he's only talking about the red circle.
That's pointing at the red circle.
Yeah. No, but so it's conjugated with glycine, so this is still technically a bile salt. But he's saying of the bile salt, this is the bile acid before it gets the glycine. Does that make sense? So bile acid,
bile salt has this.
and it's still, this is still technically a bile salt, but he's saying this is the bile acid. That's the bile salt. Once the glycine's added, and that's why I was like, glycine makes a bile salt. So why is that bile salt? But he's specifically talking about that structure, the steroid. I was like, that's it.
It was a little confusing. I was like, what are we talking about here? Where are comfort cells from?
In the hepatic pool system.
The liver. And what? And what?
Like the artery, the hepatic artery, arterioles,
You're close. Sinusoids.
sinusoids. You got above one first. It's flying.
It's funny because it looks don't need that. Where's that higher dose?
You got time.
I wonder if that would help. That'd be nice.
Oh, it's awesome. Damn voices. Let's go.
All right, guys, let's get started back. We are shifting gears, and now we're going to be talking about the methionine cycle, the folate cycle, and how vitamins are involved in these pathways. So we know methionine is another essential amino acid. Folate is vitamin B9, and so these pathways, the methionine cycle is very important, methionine and folate cycles, because they are involved in cellular development, growth, DNA synthesis, protein metabolism. We're going to talk about examples of each. And so, just as a little prelude, if we have any deficiencies or alterations in these pathways, it's going to automatically affect our DNA and protein synthesis, and that's going to be manifested in some of the side effects that we see. So, methionine is very important in methylation. Methylation is the adding one carbon group to DNA or proteins, and we're going to talk about how it does this. But we know methionine contains sulfur group. What's the other amino acid that contains sulfur? Cysteine. Perfect, cysteine. And we're going to talk about that as well, because methionine can be converted and is sometimes converted to cysteine in what's called the transsulfuration pathway. And so there are four main substrates in the methionine cycle we're going to talk about, and methionine relies on cobalamin, which is vitamin B12. So if there's a vitamin B12 deficiency, we're going to have issues with the methionine cycle and the folate cycle. But so what happens is we We have to consume methionine. When we do, it is converted to S-adenosylmethionine, which is the universal and major methyl group donor for the body, by methionine adenosyl transferase and ATP. So what does methionine adenosyl transferase need to work? ATP. Okay. So that SAM is then converted to S-adenosyl homocysteine, SAW, and that occurs by methyltransferase. SAW is converted to homocysteine. Homocysteine is very toxic to our body. We're going to talk about what it does a little bit later on. But this reaction, SAW to homocysteine, occurs via S-adenosyl homocysteine hydrolase. Then that homocysteine can go down the transsulfuration pathway to produce cysteine, or the homocysteine can regenerate methionine. And we're going to talk about how it does that. Of importance, this pathway, this transsulfuration pathway, we're going to talk about some disorders when enzymes are deficient. These enzymes are in red. One thing to note that vitamin B6 is a cofactor for this pathway. So one way to treat this issues with enzyme deficiencies is going to be vitamin B6. Right? So we will talk about that. Here are the key steps to the methionine cycle. The main step being ATP is required to convert methionine to S-adenosyl methionine. Right? So, if there are deficiencies in any of the enzymes in this pathway, particularly we're going to talk about the transsulfuration pathway that I showed at the bottom. They are going to cause methionine metabolism disorders. First one we're going to talk about is homocystinuria. This is the result of cystathionine beta synthase deficiency. And so what happens is this homocysteine, or this methionine pathway is very involved in development of connective tissue. So when there's a deficiency, you're going to see symptoms that are involved in connective tissue. Osteoporosis, scoliosis. Also, homocysteine is toxic to the central nervous system. That's why you see the intellectual ability. Homocysteine is also toxic to our vasculature, our blood vessels, so you have the increased risk of developing an embolism. So we treat this deficiency with vitamin B6 and other vitamins as well, vitamin B9, folate, and also betaine. Betaine is essential amino acid, it's an amino acid derivative. It's related to glycine. So it helps in methyl donation, and it will treating a patient that has cystathionine beta synthase deficiency. This is one of the considerations that you might try. And of course, you would decrease the diet of that, so it does not go down this pathway. Another condition, homocystinuria with megaloblastic anemia. So the enzyme that is deficient is not directly in the transsulfuration pathway shown here, but it's off to the side. It's up here, and we're going to talk about how it works. So here is the methionine cycle here, methionine at ATP, S-adenosyl methionine, SAM, SAW, and then here's homocysteine and the transsulfuration pathway. So in order to regenerate that methionine from homocysteine, this needs to occur by a reduced vitamin B12. The enzyme that's responsible for ensuring vitamin B12 is in the correct form, that's able to work with methionine synthase is methionine synthase reductase. So as you can see here, this methionine synthase reductase is basically shuffling out the vitamin B12. It's getting it in its reduced form so it can go on to be used to convert homocysteine to methionine. So when this enzyme, methionine synthase reductase, is deficient, again, you're going to have intellectual disabilities because homocysteine is toxic. You're also going to have issues with blood cell, this megaloblastic anemia. This is due to, remember I said the methionine cycle is very involved in DNA synthesis, so it affects the synthesis of red blood cells. It dysregulates that, so it basically makes the process of cell division and growth go out of whack. So then that's going to cause basically immature large red blood cells, megaloblastic anemia. And you would treat with vitamin B12 and methionine and also folate vitamin B9. Any questions? Another enzyme deficiency, and this was part of the transsulfuration pathway, it's cystathione gamma lyase, and it causes cystathionuria. What that means is that we have an excess of cystathione here because this enzyme is deficient. So fortunately, cystathione is not as toxic as homocysteine. So you might see intellectual disabilities in severe cases, but most of the time the person is normal, and you would treat that patient by giving them vitamin B6. Questions? All right. Now the folate cycle. So when I think about the folate cycle, I think about what I learned in school like 30 years ago, that folate is deficient, it causes neural tube defects. That's the only thing I remember from school. But, and it does do that, and we know folate is found in green leafy vegetables, also nuts and stuff like that. Folate supplementation is also used to fortify our food, but we don't absorb it as well, so it's not doing all that much sometimes. But when there's a folate deficiency, it too is going to cause that megaloblastic anemia that I mentioned, and neural tube defects, because again, it's affecting DNA synthesis and protein development as well, in addition to RNA synthesis and metabolism. So here are the key steps to the folate cycle. Now, I'm not going to lie, these names they're a pain, but make sure you guys know the difference in these names. Some of them are close. Well, these two are close, but we're going to talk about it. So what happens is the dietary folate, we take it in our diet, it's converted to dihydrofolate by dihydrofolate reductase. DHF is converted to tetrahydrofolate. Tetrahydrofolate is the active form of folate needed for DNA synthesis. It goes on to make thymine, the nucleotide thymine. It's also used in methyl donation, that one carbon methyl group donation, and in amino acid metabolism. Then that's converted to 10-formyl tetrahydrofolate, which is then converted to 5,10-methylnyl tetrahydrofolate to 5,10-methylene tetrahydrofolate. Right. Riboflavin is important in this reaction, and there's something different about this one. I'm not going to say what it is because you guys can see. But this is very important because this 5-methyl tetrahydrofolate, because it is the substrate that marries the folate and the methionine cycles together. this molecule will donate a methyl group to homocysteine in the presence of vitamin B12, or with the help of vitamin B12 to regenerate that methionine. Now, my predecessor, he had another name for this acronym. You can probably guess what it is. I can't say it, but just maybe it might help you guys remember it. Maybe just about. You don't have to take it. No, not getting got. Any questions about this? All right.
I got a question.
Yes, I'm sorry.
So according to the folate cycle, what are the key takeaways here? Did I highlight it?
Key takeaways are, that's not a bad question. So, key takeaways, tetrahydrofolate is what? Active form of folate. That 5 is, right, is what? What does that 5-methyl tetrahydrofolate do?
Methyl.
Methylation, absolutely. So those are the key takeaways. And so how it does it is you can see the 2 cycles, the folate cycle here, methionine cycle here. That 5-methyl tetrahydrofolate will donate that methyl group to homocysteine with vitamin B12 to regenerate methionine. Does that make sense? Yes.
Sir, after the 5 MTHF donates that methyl group, does it just recycle it?
Yeah. Any other questions? Yes.
Sir, for the unmetabolized folic acid, does it just not go through this process?
We eliminate it. Yeah, it doesn't get absorbed well, so we just eliminate it. Any other questions? All right, and so deficiencies in folate. Of course, we talked about the megaloblastic anemia. Because this cycle is involved in DNA and synthesis and protein metabolism, you're going to see a number of conditions associated with anemia. So our red blood cells are going to not develop properly. That's going to cause less oxygen to be transported through our body. So you'll see heart palpitations because our body's trying to pump blood more to get oxygen to our tissues. Neural tube defects, as I mentioned, that's one of the things that we commonly remember. So you would treat folate deficiencies with vitamin B supplementation in general. Of course, folate, vitamin, and then other B vitamins because they all work together. Vitamin B9, 12, and 6. So now there are drug interactions with folate deficiency. Methotrexate is a cancer therapeutic drug. It is an antagonist. Methotrexate and trimethoprim. Trimethoprim is an antibiotic. Those block dihydrofolate reductase. So basically, we're not able to start this pathway at all. So we don't get dihydrofolate and we can't continue this pathway. That's what methotrexate and trimethoprim does. Oral contraceptives and 5-fluorouracil and the anti-seizure drugs, carbamazepine, phenytoin, valproate, they all alter folate absorption in our GI tract. So we're not going to get intake enough folate when we're taking these drugs. Any questions about drug-drug interactions? Yes.
Another question about the drug interaction, but maybe I missed it. There's not a rate-limiting enzyme in the folate cycle, is there?
That's not a good question. No. Good question. Any other questions? Typically, if there's a rate limiting enzyme, I will put it on there, but just check me. You can always ask. I want to make sure that's happening. Right? This is that megaloblastic anemia that results due to vitamin B12 or B9 deficiency. That's going to cause, it's going to alter basically the cell cycle. So you're going to get very large immature cells, red blood cells, and also you're going to get hyper-segmented neutrophils due to the same reason the cell cycle is altered. Here are the neutrophils, and here are the megaloblasts, the large immature red blood cells. All right? Continuing on with molecules that are important for the blood, heme synthesis. So heme synthesis, this pathway is a multi-step pathway that occurs in the bone marrow and liver, and it occurs in multiple organelles. So it occurs in the cytosol and the mitochondria of those tissues, with the goal of producing heme, which we know is a component of our blood. Heme is also very important because it is a prosthetic group that's found in a number of our enzyme systems, to include the enzymes involved in the electron transport chain and the cytochrome P450 enzymes. So cytochrome P450 enzymes contain heme as a prosthetic group. Here are the key steps to heme synthesis, and we're going to walk through it. So succinyl-CoA, this is why I wanted you guys to know when succinyl-CoA was important, why it was important. So that and glycine are the substrates to make delta aminolevulinic acid. That occurs by ALA synthase and vitamin B6. This step occurs in the mitochondria of bone marrow and liver cells. That delta ALA will leave the mitochondria enter the cytosol to become porphobilinogen. That occurs by ALA dehydratase. As you can see, ALA dehydratase and ferrochelatase, which is the final enzyme in this pathway, are inhibited by lead. We're going to talk about the effects of that. But this porphobilinogen is converted to hydroxymethylbilane and uroprofrenogen 3. Now, this step is catalyzed by the deaminase enzyme here. When this enzyme is deficient, it's going to cause acute intermittent porphyria. We're going to talk about some of the hallmark side effects and symptoms of that. This uroprofrenogen 3 is converted to coporphrenogen 3 by urod decarboxylase. When that is deficient, it causes porphyria cutanea tarda. This substrate or intermediate will then re-enter the mitochondria, become protoporphrenogen 9, which in the presence of iron added to it, and ferrochelatase will become heme. It's a lot of words and a lot of molecules.
Who made those words?
So
draw that out.
we're going to talk about what happens when there's deficiencies in these enzymes and when lead interacts with two of these enzymes. So lead poisoning is a result of its blocking ALA dehydratase and ferrochelatase. And so here are some of the main symptoms that you will see that will be determined in the lab. The ring sideroblast. These microsideroblastic anemia cells are basically, there's too much of the intermediates, excuse me, sorry. So this enzyme is, when lead blocks this enzyme, this builds up, delta ALA will build up into the cells, and that's gonna inhibit heme production. So you're gonna have small, immature red blood cells that are overloaded with iron. So that's why you see the dots, which are representative of iron molecules in the cell. You also see Burton's lines. Burton's lines, this doesn't always occur during lead poisoning, but basically that's a lead deposition at the interface of your teeth and your gum line, as shown here. And then the basophilic stippling occurs because basically there's intermediates of this pathway that build up in the red blood cells. And so, of course, this is common, lead poisoning is common in older houses. A lot of paints included lead. So that's one thing that is now required when purchasing a home. You have to know if lead was used in the paint or any other product in the home's development. Now also what will occur is you'll have increased aminolevulinic acid in the urine because it builds up, so much of it builds up in the urine, it spill over into the urine. So when that porphobilinogen deaminase enzyme is deficient, you will see acute intermittent porphyria. So this is serious because those porphyria precursors, the intermediates, are very toxic, and so they can cause a number of symptoms, including anxiety, paranoia, and depression for one, and that's because they are toxic to the central nervous system and they interfere with glutamate and GABA neurotransmission, which are very important in our central nervous system. Also, the porphyrinogen substrates or intermediates, they're the color of port wine. So you can see here normal urine and AIP urine, it looks like wine. And then you might experience, or your patients might experience, severe abdominal pain, and that's because the nervous system, the enteric nervous system in the gut is targeted, and so that's going to give them severe pain potentially. So they might have nausea, vomiting, or diarrhea. Any questions? And of course, they're going to have a backup of the previous substrate before the enzyme, which is porphobilinogen. Now for PCT, or porphyria cutanea tarda, this occurs when there's a deficiency in UROD3 decarboxylase. And as you can see, it causes significant hyperpigmentation, and upon exposure to UV light, it causes free radical formation. You see, so the skin is significantly damaged. Like there's depositions of the intermediates here. And so drinking alcohol with this condition is not recommended because alcohol consumption will increase the production of cytochrome P450 enzymes. And as I mentioned, cytochrome P450 enzymes use heme as a prosthetic group. So you're going to basically continue with the heme synthesis pathway, but because it cannot be completed, you're going to get a toxic buildup of the intermediates. Does that make sense? And then the intermediates, unlike the previous issue, the intermediates are blue, or excuse me, orange, reddish looking, and so the urine will look orangish and red in this condition. Any questions? Yes.
Would that present as brown rat or like?
Sorry, brown. It looks kind of orange red to me. All right. Any other questions? All right. heme degradation. So this pathway, we're gonna go over it just so you guys can see it. It's of course it's significant, but you know I wouldn't focus your future studies on it that much. So this pathway basically is trying to remove the heme from red blood cells. Red blood cells live about 120 days, so that hemoglobin is released from the red blood cells, and then our reticuloendothelial system in our liver and spleen and macrophages will basically convert that heme first to biliverdin, and the colors correspond to what the actual substrates or intermediates look like. Biliverdin, this biliverdin is not water soluble, and it's converted to bilirubin, which is also not water soluble. So in order to get it to be able to be excreted, it is conjugated via the enzyme UGT1A1, and that produces a bilirubin that can leave and be excreted in our feces. Questions? All right, we're almost done, guys. And now we're going to shift, still talking about blood, but now we're going to talk about coagulation. So we know coagulation is our body's mechanism of trying to stop bleeding. And so there are two main ways. We have primary hemostasis, which our body forms temporary unstable platelet plug. Secondary hemostasis, where our body forms a stabilized fibrin cross-linked plug that's more stable and definitely with the goal to stop bleeding. We're going to focus on the secondary hemostasis. And so, again, this is just talking about three mechanisms to stop blood. And our focus will be on the blood clotting cascade, which is secondary hemostasis. So we know this pathway involves, it's three pathways that come together, two pathways that come together in a common pathway, and the goal is to form a stable clot. So we're going to talk about some deficiencies, that what happens when there's deficiencies in some of the clotting factors. There's about 12-ish clotting factors. So just so you guys are aware, when you see clotting factor with an F, you may see it with the F, maybe without an F, but when it has the A next to it, that means it's an activated factor. And without the A, it's the zymogen, which is the inactive form of that clotting factor. So here is the coagulation cascade. We're going to talk about each arm of the pathway and how they come together. First, starting with the intrinsic pathway. So what happens in the intrinsic pathway, we're going to talk about it in order of the clotting factors as they go down the pathway. So what happens is factor 12 is normally floating around in the blood when it's inactive. But when there's vascular injury, collagen from the subendothelial layer, which is negatively charged, will be exposed. And so that factor 12 contact with collagen will activate that factor 12, which is also Hagman factor, shown here. And that will begin the intrinsic pathway. So this factor is the only factor in all of these pathways that, if deficient, does not cause any blood clotting issues. Okay? So once factor 12 is activated, it will then in turn activate factor 11. Again, we're in the intrinsic pathway still. So when factor 11 is deficient, that is hemophilia C. Hemophilia C is a mild form of hemophilia where the blood issues are only occur, particularly like post-surgery, and we're gonna have a little chart at the end that gives just a brief snapshot of the differences between the different hemophilias. So factor 11 is activated, and then it go on to activate factor 9. Factor 9 is the Christmas factor. This factor was discovered in Great Britain in a little boy with the last name of Christmas. That's how it got its name. This is a vitamin K dependent factor. There are 4 of them. We're going to talk about all 4 of them. When it's deficient, it causes hemophilia B. Hemophilia B and C, or excuse me, hemophilia C causes minor bleeding. Hemophilia A and B cause major bleeding, and they are more serious. So once factor 9 is activated, that will activate factor 8. Factor 8 consists of 2 factors or 2 variables, factor 8 and von Willebrand factor. Those complex together. The von Willebrand factor is involved in platelet support for platelet aggregation, and it's involved in primary hemostasis as well. So when factor 8 is deficient, this is where we see the classical hemophilia that we know, hemophilia A, which is X-linked. Both hemophilia A and B are X-linked, meaning that they're found primarily in men. Factor C, I did mention, is found in a subset of the Jewish population in males and females. So now, factor 8, once it's been activated, were there any questions? Factor 8, once it's been activated, it will then activate the Stewart-Prowler factor, which is factor 10. This now enters the common pathway, and it's also vitamin K dependent. All right. So now we're going to jump up to the extrinsic pathway. So what happens in this pathway? Tissue factor 3 upon vascular injury. Tissue factor 3 is not typically exposed to the blood. So what happens when there's injury, it comes out of the endothelial layer of the blood vessel. And so that activates it, and then it begins the extrinsic pathway. So it will complex, factor 3 will complex with factor 7, which is also vitamin K dependent. It also complexes with the other components that are circled here, and we'll talk about them in the next slide, to activate factor 10 and the remaining steps of the common pathway. Right? So once the common pathway is activated, factor 5, factor 4, which is calcium, and lipids. Lipids are phospholipids. Serine and phosphatidylethanolamine, they all complex together to help form the necessary assembly in order to activate prothrombin, which is factor 2, and prothrombin is also vitamin K dependent. And then prothrombin, when activated to thrombin, it will activate factor 1 fibrinogen, which will then activate factor 13 and stabilize that fibrin clot. And these are the steps to the secondary hemostatic clotting cascade. I know this is very important, guys. I'm sorry. Any questions?
So does this cascade has to follow this order?
Yes. Yeah. In the sense that it's following the orders in the sense that these must be activated, like tissue factor activates this, which activates this, or the factor 12 activates factor 11, activates factor 9, activates factor 8. That's the order it follows. Does that make sense? Yes.
I don't know how to word this, but I guess like what makes it extrinsic versus intrinsic?
That's a good question. No, very good question. So, and thank you. So, intrinsic pathway, the intrinsic pathway is essentially like due to when we have an internal injury. For example, if we have like severe inflammation that somehow ruptures our vasculature, that's occurring internally. So, intrinsic is already occurring internally. It's going to disrupt the vasculature. Extrinsic would be like if we were stabbed or if we have surgery, it's coming from outside. Does that make sense? Thank you for that question. Now, the vitamin K dependent factors are 2, 7, 9, and 10, shown here. And here is a summary of the clotting factors. What you guys need to know, what I would recommend is just knowing how the order of how the cascade pathway works, and then that'll kind of tell you what's going on in the pathway. Like, I don't expect you to remember all the words and all this stuff. It's a lot of words, but I would recommend you guys learn the different names of the factors if there's one associated. Any questions? Yes.
I thought that there were 12. There's 13 of them. Isn't that 13?
One of them is missing. So there's 6 is not there. So what happened was they thought there was a factor 6. And so, but then they realized factor 6 was another molecule that wasn't really a factor on its own. So, but they continued on with the other number. So it ends up only being 12. Yeah, they didn't want to renumber. You know how they do. They do stuff make it difficult. So when we're going through it, we're trying to figure out what's going on, and we can't find 6. We don't know what we're doing. Well, that's how I am. I'll be like, wait a minute, where's 6? I don't remember about 6. So, yeah. Any other questions? And then this is just for your FYI, the different forms of hemophilia. A and B are severe. They're found in men, excellent. And then C is found in the Ashkenazi Jewish population, and it's mild. All right? So, if you had a patient with blood clotting issue, how would you treat them? Say again? Give them the factor, yeah, absolutely. Yeah. What, somebody said something else?
What there?
Keep them from getting shot, thank you. That's good in general. Yeah. I think they will appreciate it no matter. I would, don't get shot, don't get got either. Same thing. Yeah. So, good point. So, you would treat your patient with heparin. Heparin targets the intrinsic pathway, or warfarin, coumadin, and that targets the extrinsic pathway. The test that you would use is activated partial thromboplast time to measure the progress or how well the patient is doing on the drug, and that the APTT is measuring, basically, essentially, it's measuring the speed of the intrinsic pathway factors. Prothrombin is measuring the extrinsic pathway. And so, what I want you guys to take home message for that is that, so heparin is used inpatient. It's for like emergencies. It's for deep vein thrombosis or pulmonary embolism. So you would be treating a patient that has those issues emergent because they have cardiovascular issues, or if they're undergoing a surgery and you want to make sure that they don't develop clots. And so DVT and pulmonary embolism, and this is given IV. again, it targets primarily heparin targets the intrinsic pathway. Now, warfarin targets the extrinsic pathway, but the focus is on the vitamin K dependent factors, which are 2, 7, 9, and 10. Or sorry, right? 2, 7, 9, and 10. Yes. And so how it does this is that warfarin basically blocks the recycling of that vitamin K. Just like in that condition that we saw, the methionine, this is the homocystinuria, that methionine reductase enzyme was shuffling the vitamin B12. Warfarin blocks the shuffling of vitamin K, so it cannot be used. So these factors will not be able to aid in producing a clot. And so this drug is used orally, so the patient will take it. And this drug has a what's called a narrow therapeutic index. We're going to talk about that in the last plot. And so you have to monitor them very carefully. They will have to come to the lab to get frequent, regular blood draws to ensure they're compliant and they're taking the medicine as needed. Because you don't want them to take it in a way that will cause an increased amount of drug in the body, because that's going to mean that they can have some potential life-threatening effects. All right, and so this is used primarily for DVT development and pulmonary embolism as well, but it's also used for patients with mechanical heart valves. You don't want them to develop a clot. All right. Any questions about this lecture? So the other ones are not this bad. This one, I will admit, it's bad for me to stand up here. I'm sweating. I feel like I'm on trial. So, but as you guys, as we go along, if you have any questions, of course, I'm here to assist. And I want to mention, if you have questions, please email me through Canvas, because those questions come to my personal email. So I don't check my work email all the time when I'm out of the office. So I just want to make sure I do answer your questions. So if you guys have any questions, in my office, 1319. But I'll see you guys tomorrow at 8. All right. You guys have a good afternoon.